different topic than what we planned earlier. Suggestion you people have had that you go for sale and cell biology first because it plays very important role in all the aspects and therefore it's always good study it anyway. So we'll go for that. Now the syllabus asks for it starts with cell biology and various organelles, various macromolecules, etc. Basically, all these things you have studied at various places. You have studied it in physiology, you have studied it in molecular biochemistry, you have studied it in microbiology, and definitely you have studied it in 11th and 12th as a part of biology, botany, and zoology. So, most of the things you have studied, only you need to add up some more details, and second thing is you need to uh, revise the things which you have seen earlier. So when we go for that, we know cell biology. What is cell? You know. So cell is the smallest unit which comprises the organism. We can have a variety of organism. Can be unicellular, it can be multicellular, and accordingly, if it is unicellular, then cell is everything for that because it is a single cell. If it is multicellular, then cells combine together. If the organism is a bigger one, then the cells with similar function come together to that what we call them as the tissues. Then these tissues come together to form having a similar function to what we call as a organ and organ functions. That all you studied in physiology. But yes, what we understand is the cell is the basic structural and functional unit of the body. It's a very simple definition that gives clear information about what cell is doing and what cell has to do, that is structurally and functionally. It has to play the role whatsoever been designed, whatsoever been assigned to that particular cell, if it is unicellular, or for that particular group of cells or tissue or organ, whatsoever it may be. So this is a structural and functional unit. Basically, these are divided in important divisions. So these are we call them as prokaryotes and eukaryotes. You might have studied this also. So these prokaryotes are particularly for single cellular organisms. These prokaryotic cells are there. In eukaryotics, these are the cells which are comprising multicellular organisms. So in that there are bacteria, archaeas. They are the organisms which are having prokaryotic cells, while animal, plants, fungi, and the bigger organisms they have eukaryote cells. The differences are the size is 1 to 5 micrometer in case of prokaryotes, while in eukaryotes it can be ranging from around 10 approximately to 100 micrometers. Type of nucleus, it has got only nucleoid region, it doesn't have any two nucleus, while in eukaryotes it is having a two nucleus with double membrane, just like a cell membrane. We have got a dual membrane or fluid mosaic model which we see. So similar kind of it is there. The prokaryotes having a DNA which is usually a circular DNA, while in eukaryotes the DNA is of linear molecules. That is, their chromosomes and their chromosomes are wound on the histone proteins. We shall be seeing all these details in future one after another because these are the part of the further syllabus. Then RNA protein synthesis, how it is happening? It is coupled in the cytoplasm. Okay, this these are coupled in the cytoplasm, and the RNA synthesis is taking place in eukaryotics. If we see, the RNA synthesis takes place in the nucleus, while the protein synthesis in the cytoplasm. Now, all these things actually you know basically RNA synthesis, protein synthesis, you know that what happens in this nucleus, that is what all the process you have seen, that is transcription, how it is taking place, transcription from DNA to RNA, then the next stages of RNA, its splicing, etc. I'm just giving this word because I'm very sure many of you are not prepared, despite we discussed that some of the basic has to be prepared. But still, I'm reminding these words. You have seen all this particularly that how RNA synthesis takes place in human cell. That is, it takes place in nucleus. We call it as a transcription. Then these RNAs they come out. This transfer RNAs. Then they bind with this messenger RNAs. Then they bind with the ribosomes, and then the ribosomes are present in the cytoplasm. And they do it. So this is how we have seen. Again, ribosome structure we can see in more detail as we go for organelles. Basically, the difference in prokaryotics and eukaryotic is 50S and 30S. These are two subunits which are present in the ribosomes of the prokaryotics or bacterial or archaic cells, while in human being it is 60S and 40S. So these are the again very important one. This is the basis of mode of action of most of the antibacterial agents, which we shall be seeing when we shall see chemotherapy. That why they are more selective, because these are 50S and 30S, while in human beings it is 60S and 40S. So because of this structural differences, uh, this particularly giving the specificity. Then cytoplasmic structure in cytoplasm comparatively in prokaryotes very few structures are there. While in eukaryotics it is highly structured by endomembranes and cytoskeleton. Again, I hope you can remember the cytoskeleton if you are remembering the cell division. In that, how cytoskeleton plays microtubule and microfilaments, they play important role. And endomembranes are there, which are segregating various organelles. So these are important divisions which are there between cytoplasmic structures. Cell movements in certain prokaryotic flagella is there, and they are made of typical protein that is called as a flagellin. In eukaryotic flagella and cilia, they are containing microtubules. They are having lamellipodias and phyllopodia containing actin. So these are such variety of proteins which are involved in that. Then in prokaryotics, mitochondria are not seen, while in case of eukaryotics, there are uh, minimum one to several thousand mitochondria can be present depending on the cell and its function. Chloroplast is not present, while chloroplast is present in algae and plants in eukaryotes. Organization usually it is a single cell, as we have seen it is unit. The organization is usually single cell, while eukaryotes can be single cells, very few colonies. Then higher multicellular organism with specialized cell to what we call them as a tissues and organized form as organs. How the cell division takes place in this book. In cell division, it is see all prokaryotic cells you have studied in microbiology. Okay, we are just summing it up. I already told you to go through this. So these are three basic sciences where you have studied about cell. I am repeating. Okay, so this is where you have studied is these are cell division. It is binary division, that is simple division. While here we have in the eukaryotes, it is a mitosis and meiosis. Mitosis is fusion or budding, and meiosis again it involves uh, many other processes like crossover, etc. Okay. Then chromosomes in prokaryotics, it is a single chromosomes, while in eukaryotes, it is more than one chromosome. In membrane, there is a single cell membrane, while here it is cell membrane and membrane bound organelles, and in plant again it is plant cells are there. So these are a variety of uh, uh, differences are there in prokaryotic cells and eukaryotic cells. So I am sure you are just recalling, you are able to recall the things which have been there. Now, uh, if we go ahead, this is all about cell. What are the cells? Which are the different? Uh, what are the differences between eukaryotes and prokaryotes? This is a simple cell, uh, what we can say, classification. Now, when we say about subcellular components, or when we go for the differences in variety of cells, the first thing is there, that is cell membrane. Now, again, you have studied cell membrane in details. You have studied it as a fluid mosaic model, which is very commonly called as, and include last year when you might have studied absorption, pharmacology one. So you have studied it as a, there is a fluid mosaic model is there where it is linked by layer, etc., etc. So this double layer of phospholipids is there, then polar, non-polar, or amphiphilic, lipophilic type of, or hydrophilic, lipophilic heads are there, polar heads and non-polar heads are there, or polar head and non-polar tail we can call. They are there, and there are some pores, some what we can say channels are present, and thus it is helping in the various cell-related activity, membrane-related activity. There are membrane-bound receptors are there. So we have seen most of the things earlier as a physiology. Okay. Then next important part in that is the cytoskeleton. We shall be again discussing it at various places when we shall discuss about cancer also. That plays important role. This cytoskeleton, cyto means cell, skeleton means skeleton, just like our bones are there. They are providing skeleton, and because of that, it is providing a very good framework, and which is responsible for the strength, uh, size, shape of the body. To know what are various function of a skeleton. Similarly, this cytoskeleton that gives you the maintain cell shape. It anchors organelles in a place. It helps during various processes like endocytosis, that is the uptake of material and cytokines, then separation of daughter cells after cell division, then move the part of cell in progress of growth and mobility. There are many functions that it is carrying. The eukaryotic cell cytoskeleton is composed of microfilaments, intermediate filaments, and microtubules. Again, we shall study this as a part of anti-cancer, because we know there are certain agents which are acting on microtubule, and because of their action on the microtubule, they will not allow microtubules to form, and because of that, the cell division will be affected, and no cell division means it will arrest the cancer. So there are certain drugs when we shall study in cancer, we shall be studying. So there are great number of proteins that are associated with them, each controlling cell structure by directing, bundling, aligning filaments. While in prokaryotic it is less well studied, but it also involves the maintenance of cell shape, polarity, and cytokinesis. Cytokinesis, again, this term we have seen cytokines earlier. Here cytokinesis is there. So cyto is again cell, kinesis is a movement. Movement again, it can be inside the cell or outside the cell. Okay, so these are playing important role. The subunit protein of microfilaments, they are small monomeric protein, they are called as actin. Subunit of microtubule is a dimeric molecule, that is called as a tubulin. The intermediate filaments are heteropolymers. Their subunits are along with the cell types in different tissues. Some of the subunit proteins, they are they are known as a Vimentin, desmin, lamin. Again, lamin, ABC, keratin. Keratin, you must be aware about keratin, you must have heard. Keratin is part of the outer epithelia, that is the squamous epithelia of skin, and the keratin is the major component of the skin, or we call it as a dead skin, hair, nails, etc. So this is there are multiple acidic and basic keratins. Neurofilaments presents are present like NF, neurofilament L, neurofilament M. Okay. These are various things are present. These are we are studying in brief. We are doing a very fast review of the things. Then next is genetic material. There are variety of genetic material we have seen in form of DNAs, RNAs, and this particularly arranged in a typical way. Then we have got a variety of versions of RNA. Example, we have seen for information transport is a messenger, for enzymatic function like ribosomal, for transferring the molecules to add amino acids. They are the uh, transfer RNA, and thus they are playing important role. Each RNA is playing important role in protein synthesis. Again, I hope you might have gone through protein synthesis well. While prokaryotic genetic material is organized in simple circular bacterial chromosomes, it is present in the nuclear region of the cytoplasm, because we have seen that it is not very clearly demarked the nucleus. Eukaryotic genetic material is divided into different linear molecules which are called as chromosomes. They are present in the discrete nucleus. They are usually having additional genetic material, and some of the organelles like mitochondria and chloroplast, they are present into that. In human cell, because we have to go in more detail about this later in later section of the last section of this, that is having a genetic material. It is present in the cell nucleus. We call it as a nuclear genome. It is also present in the mitochondria. We call it as a mitochondrial genome. Then humans have a nuclear genome that is divided into 46 linear DNA molecules. They are called as a chromosomes. They are having 22 homologous chromosome pairs and one pair of sex chromosomes. So it total makes 46 chromosomes, 22 pairs, and one is sex chromosome. The mitochondrial genome is circular DNA molecule. It is distinct from the nuclear DNA, and it is very small compared to nuclear chromosome, but it codes 13 proteins that are involved in the mitochondrial energy production and specific transfer RNAs. This is very very important feature we have to think about, because mitochondria are not depending on the uh, these chromosomes and genetic material to do the things, because the energy production is a very very continuous process. So the physiologically this type of feature is there, where mitochondria they themselves are enabled for synthesizing their own enzymatic machinery for energy production. Now, foreign genetic material can also be there, which can be artificially introduced into cell body, and that process is called as a transfection. This we shall study again in further section. We call it as a gene transfer or gene therapy. It can be transient if the DNA is not inserted into cell's genome, or stable if it is inserted into the normal one. Certain viruses are also insert their genetic material into genome. Again, this we shall study when we shall study antivirus or drug. You must have been studied virus, that how this virus are doing, or how viruses are infected in microbiology. That and particularly now this in COVID situation, those who might have having habit to read the things, that how viruses are doing. Viruses are attaching, they are inserting their protein into cell, then that this genetic material will cause production of proteins. So this is that is what the genetic material is. This is a foreign genetic material human cells are getting, and because of this foreign genetic material, the human cells they are processing it without understanding it is foreign, and thus they are making copies of viruses. Okay, so this is how the differences in prokaryotic, eukaryotic, and viruses and all about. Then next is organelles. Uh, Again, there are variety of organelles. We shall be studying it separately as a what organelles are present and how they function. Because I'll just make the list which you might have studied earlier. Uh, then all various structures which are present uh, outside the cell. So we shall go for the structure which are present outside the cell membrane first, and then we shall go for organelles in the next, because next will be organelles. Okay. Now we have seen already that there are cell membranes are present. Call it as semi-permeable cell membrane, and the component must be carried across the cell membrane by process. So what's the function cells are doing? For that, the transport is very very important across this cell membrane. There should be the nutrients should come in, and the products may it be waste product or may it be functional product like hormone, enzyme, or any neurotransmitter, whatsoever it may be. So it should be come out of the cell where it is made. So this is how this should occur. So there are cell wall. It is present in most of the prokaryotic, eukaryotic cells. They have cell wall. Uh, Now, different type of organism, they have different kind of structure. It acts to protect the cell mechanically and chemically from the environment. The additional layer of protection to the cell membrane. So it is addition to the cell membrane. I repeat. So this is the different type of cell. They have different cell wall made up of different material. Plant cell walls are primarily made of cellulose, fungi are made up of chitin, and bacterial cell wall are made up of peptidoglycan. Human don't have cell wall; they have only cell membrane. Okay. Now this again this gives a very very important modification. So we can have various drugs which are specifically acting on chitin. There are specifically we have drugs, example penicillin, etc. They are acting on the bacterial cell walls, so that since human does not have cell walls, we can have selective action on the bacterial cells. So this is how this structure or the differences are there. Then in prokaryotic, some of these are having capsules. That is, a gelatinous capsule is present in some bacteria outside the cell membrane and cell wall. This capsule may be having polysaccharides. Example of these are pneumococci, meningococci, or polypeptides like Bacillus anthracis. Or it can also have an enzyme that is, it can also have a typical material that is hyaluronic acid, which is there in streptococci. They are not marked by normal staining protocols and can be detected by India ink or methyl blue. They will allow the higher contrast between cells for observation. So in prokaryotic capsule is another thing which is present. Then next is the flagellas are present. I'm very sure in microbiology you might have seen this flagella or motility under microscope, and how flagella is playing important role. So these are the organelles for cellular mobility. The bacterial flagellum stretches from cytoplasm through the cell membrane and extrude through the cell wall. They are long and thick thread-like appendages, protein in nature. Different type of flagellum is found in archaea and different are in eukaryotes. Then fimbres are there singularly when we call it as a fimbria. It is more than one, we call it as a fimbri. They are also known as a pilus, or if it is in plural, we call it as a pili. It is a short, thin hair-like filament which is found in the surface of bacteria. They are form of a protein. They are called as a pilin, which is antigenic in nature, and they are responsible for attachment of bacteria to specific receptor on human cells, and that particular attachment to the specific receptor on human cell, we call it as cell adhesion. There are special type of pilings that are involved in the bacterial conjugation. Now I am very sure you have studied this method of conjugation and all, how the genetic material is transferred. These are various techniques in these are various what we can say mechanism processes that are involved in the bacterial this. genetic material modification process, particularly in resistance formation, etc. Now, we shall be seeing cell division separately, but we know these mechanisms. What are they? We call it as a replication. So, how this binary fission? You can see the figure in particularly prokaryotes. It is a binary or a single cell molecule. It is the binary fission that is inside the cell. There is no such nucleus or thing is there. So from that, a circular type of DNA is present, and that is causing DNA replication. And then from that, this chromosome segregation, cytokinesis, that means cells are separated from each other, and this is how the thing is there. While in case of this, the diploid cells are having various growth phases in eukaryotic cells. So mitosis and meiosis are the process they are taking place. S phase is the first phase, that is synthesis phase, and then G1, G2 phase they are taking place. I am very sure you have gone through this. Then how meiosis particularly is causing, that is haploid cells they come together, that is two haploid gametes come together to form a zygote which is diploid, and then cell division takes place. So these are all cell division processes, DNA replication, and how this thing happen. We have to see in more details in next few classes. Then important is DNA repair. Uh, there are all type of organisms. They are having enzyme system that can scan their DNA for damages, and they carry out the repair processes when the damages are detected. When this is there, the diverse repair processes have been involved in organisms ranging from bacteria to humans. The widespread prevalence of this repair process indicates the importance of maintaining cellular DNA in an undamaged state in order to avoid cell death. Because anything happens to that DNA, that means it can result to cell death because that DNA will not able to function, and we know the function of DNA is it produces important machinery in form of proteins, enzymes, and other. And these are either structural or functional proteins. And if it is not there, if any thing is happening to that, then it will result into cell death. So this to avoid cell death or errors of replication due to damages that could lead to mutation. Now you again must be knowing about mutation. We shall be encountering it in later course of part. E. coli bacteria are well-studied example of cellular organism, and it has got well-defined DNA repair process. It is having nucleotide excision repair, that is if the nucleotides are excised, they are uh, excised means they are removed from that nucleotides. Then if there is a mismatch of DNA, then this type of DNA mismatch repair is done. Then non-homologous end joining of the double stranded. You must be knowing homologous pairing. So if it is not homologous, then end joining of the double stranded break. The recombinational repair and light dependent repair, that is photoreactivation. These are various repairs. These are been notified. So these are very very important processes. By the time we go ahead, you can go through one by one for your own knowledge. We shall be seeing them at the end when we are going for further details about genetic processes. So how these are acting, how these are working, this is how important are. We are just now today going for the discussion between prokaryotic and eukaryotics. And one important thing is this prokaryotics, since they are unicellular organisms, they are easy to that way develop. We have seen it in microbiology, and it can be used. Or it provides the initial basis for the study. Particularly, it was important when the various techniques were not developed earlier. After development of techniques, various informative tools, various advanced technologies, Now people are able to, or scientists are able to go for understanding about the genetic and other cellular structure of higher organisms also. The next important thing is about growth and metabolisms. So this is again important. That is how the growth and metabolism is taking place. Very important growth. We have seen in microbiology or in other cases, growth is, we have two things for growth. Growth is increase in the size of cell is also growth after, and increase in number of cell is also growth. So, after successive cell division, cells grow through the functioning of cell metabolism. It is the process by which the individual cell process nutrient molecules. We know that metabolism has got two distinct features: anabolism and catabolism. Anabolism is synthesizing new things. Catabolism is breaking down, particularly for the better use of these molecules. Okay, so it may be for energy and other purposes. Maybe small molecules are, use example, protein are there, then we break down into amino acids, and these amino acids are used for further synthesis of different protein. So first metabolism takes place, and then new protein synthesis is anabolism. So complex sugars are broken into simple sugars. We know about everything. You have studied it in digestive system, and you have studied in various proteins, etc. So this is how the importance of growth and metabolism in cellular function. Protein synthesis is very important. Very important in what sense is, it plays very important role in cellular activity, because it is a cycle, we can say. That is, cell is there, cell is able to have various functions, because we know about cells, we differentiate or we categorize them on the basis of their functions. The functions are arrived from their activity. In activity, the proteins plays important role, and therefore protein synthesis is the, what we can say, most important functional aspect, because proteins, again, just like cell, we have got two things: structural and functional unit. Similarly, proteins are basically two types. You can have structural protein and functional proteins. So they are playing. If structural proteins are not proper, then you can imagine all the proteins which are present. We have seen just now all the which are present. May it be microfilaments, microtubules, may it be cytoskeleton protein, whatsoever kind of protein it may be. If it is structural protein is not proper, the structure will not be proper, and if it is not proper, it will not able to function that way. Functional proteins, there are many functional proteins. We have enzymes, hormones, we have got various kind of neurotransmitters, second messengers, etc., etc. So there are n number of proteins we have for each cell, and then these all proteins are important as a functional unit, and therefore a whole science is developed that is called as a proteomics. That proteomics, that involves the study of these all protein synthesis processes, various factors, and anything which is affecting it. So we shall be saying again, then we shall be going for further discussion. We shall have the details about this, a very important part for us. Then motility, we have seen flagellas, cilias, they are playing important role. In multicellular organism, cells can move during processes such as wound healing, immune response. We have seen cytokinesis. There also we have seen chemotaxis earlier, how the cells are moving and what are the signals for the motility, how various cytokines playing important role. Then in cancer metastasis, that is the spread of cancer, particularly if it's a malignant type of cancer, then how it spreads. So in that, the cells are spreading, they are moving from one organ to other organ. Then in wound healing in animals, white blood cells move. We have already seen that we call it as chemotaxis. It involves many receptors, many cross-linking, many binding, adhesion, motor, and other proteins. So it is, if we go into more depth, we'll find that there are many proteins, many receptors. many molecules are involved. And this process is divided in three steps: protection of the leading edge of the cell, adhesion of the leading edge of the cells, and de-adhesion at the cell body and particular rear portion. The cytoskeletal contraction to pull it forward. Each step is driven by physical forces that are generated by unique segments of cytoskeleton, and there are many adhesion molecules are present. They are playing important. So these are how important activities are there. Then the next things are important is multicellularity, that is in eukaryotics, how these multicellular organisms are having, then how they come together to form a similar kind of tissues, then how they are having symmetry with each other, synchronization with each other, so that they function in tandem or in one line, and how their genotypes and phenotypes are there, which are important in that. Most distinct cells that arise from single totipotent cell. We know that zygote is the first diploid cell from two haploid cells, and it differentiates in hundreds of different cells during the course of development. So all the cells which are coming from a single cell zygote, and then they derive different functions. So differentiation is very important. If something goes wrong with differentiation, then that particular functions, structural or functional aspects may not be achieved. Then, what are the origins of multicellularity? They are is present in many prokaryotes, apart also like cyanobacteria, myxobacteria, in that also it is there. But they are not complex multicellular organisms. We have only in eukaryotic they are complex like animals, fungi, brown algae, red algae, green algae, and plants. They are actually complex multicellular because so many different functions they are being done. Okay, so this is how we can have differentiation and of the cells resulting in different functions. So these are the some historic aspects are there, and origin now will not go for into more details. Okay, so this is how we completed the topics. That is first one, that is cell or cell biology cell. Now we shall see the various aspects in the due course of time. If you have any difficulties, you can put on the chat in our WhatsApp group. Okay, and keep on reading the basics which you have already read in biology, that is biology, botany, biochemistry, physiology, and microbiology. That will definitely help you, and we'll also able to make it in faster way because other topics are in. So I hope it is understanding. Take care. Be safe.